This is podcast number 7, where we're going to talk about membranes. And here is a 3D simulation of a membrane. We have the acyl chain tails and the phospholipid groups. This would be the gray on the outside would be water. So mostly a water environment facing the outside of the cell, mostly a water environment facing the inside of the cell, and a hydrophobic compartment in between. On an electron micrograph, the stain that is used in transmission electron microscopy, where you're taking a thin slice through a cell, the stain is usually uranyl acetate and also a lead stain, and those stains stain membranes. And so you get to see membranes in cells, and this is how we've learned a lot about membrane structure is from looking at these electron micrographs that you can see big membrane networks here in these 3 cells, and then you can see outer and inner membranes of of the mitochondria. In terms of membrane function, membrane is really the thing that defines the inside versus the outside of the cell. And when we talk about a semi-permeable membrane, in that some things can get in and some things can't. For example, sodium can't get in except through a pore. That's due to the function of this membrane. Membranes can also enclose compartments within eukaryotic cells, and within those enclosed compartments you have a specialized biochemistry system so that you can do reactions there that wouldn't necessarily happen everywhere in the cell. So you can concentrate. For example, you can have a low pH compartment inside the cell due to membranes that are inside the cell. And we'll talk lots about that. Membranes are the substrate through which transport happens. So if you need to get ions into or out of cells, that's how it works, is through a membrane, through proteins embedded in membranes. Cells signal each other through membranes, through embedded receptor proteins, and cells can also physically contact another cell through their membranes and protein interactions in their membranes. So much of cell function depends on membrane function. And a common misconception about membranes is that they're mostly lipid, and they're not. They're at least half protein by weight. So if you look at all these different kinds of membranes here, the lowest percent of membrane weight that is protein is 49% protein, and that's in human erythrocyte, and as high as 78% protein by weight in the mitochondrial inner membrane, where you have the electron transport chain. So a biological membrane is packed full of protein, and a lot of the function of that membrane depends on those proteins in that membrane. So please don't think about membranes as mostly lipid. They're not in most cases. There are some exceptions, but most cell membranes and organelle membranes are going to be really full of proteins. If we look at the Singer-Nicholson fluid mosaic model, so up until about the time I was probably in grade school, so me and Fred Flintstone in grade school, there were competing models of what a membrane looked like and how membranes functioned. That lost out to the Singer-Nicholson fluid mosaic model, where you have a phospholipid bilayer and proteins are embedded in that bilayer, and not only just proteins, but you also have cholesterol embedded in there, some carbohydrates embedded in there. The proteins that are embedded can go all the way through the membrane. They're called integral membrane proteins, or they can be associated with the membrane. And these integral membrane proteins have stretches of hydrophobic amino acids that can interact with the hydrophobic acyl chain tails of phospholipids. And so hydrophobic likes hydrophobic, and then hydrophilic amino acids on the uh, outside facing surface of the membrane and on the inside facing surface of the membrane. This characteristic that you need hydrophobic amino acids to cross the membrane has been used to model new proteins. So when we get sequence of new proteins, which is these days is mostly done by sequencing their mRNAs converted into DNA called cDNA. If you're looking just at a stretch of that sequence, it can be hard to know what kind of protein that is. But if you have about 20 or so hydrophobic amino acids in a row, that's a good clue that that may be crossing a membrane, because that's about the distance that is needed to cross most membranes. And therefore, you can get a sort of topographic map of where this protein crosses a membrane, if it is indeed a membrane-bound protein. The phospholipids that make up part of the membrane, again, not most of the membrane, but half or less of the membrane by weight. These phospholipids are diverse. They don't have just one kind of structure. They have one general kind of structure, but then there's a lot of diversity within that structure. So the head groups that are on the phospholipid can be different. The fatty acyl tails can be different as to what is the species of fatty acid that is attached to this tail, and they are not necessarily the same. This fatty acid can be different from this fatty acid. And all the lipids in a membrane are not phospholipids. You also have cholesterols that are also in lipids, phospholipids. If you look at just the composition of the types of phospholipids that are in different kinds of membranes, you can do this by extracting phospholipids from a cell and then subjecting them to either gas chromatography or mass spectrometry to get an idea of what are the species of phospholipids in those membranes. And if we look at the scale and say if we look at the percent of the total phospholipid and what is each kind, you'll notice that a common contender for a lot of these is phosphatidylcholine. You see a high percentage in rat liver and in potato and in humans, myelin sheath, and in spinach and in the inner mitochondrial membrane of rat liver. Another common one is phosphatidylethanolamine. You'll see these a lot. These are actually common in as food additives too. Sometimes they're called, not this big name, but another name is called lecithin, that you will see phospholipids in food. But the point is that a membrane is not going to be all one kind of phospholipid. There are phospholipids that can be modified with a carbohydrate attached to them. These are called glycerolipids or glycosphingolipids. These carbohydrates attached to phospholipid head groups can be responsible for different signaling properties of cells. For example, the human blood types, type A, type B, and Rh positive are due to carbohydrate additions to the phospholipids in red blood cell membranes. The blood type O means no carbohydrate additions, and A versus B means different types of modifications to those phospholipids, and that changes the shape of the phospholipid. And when you change the shape of that phospholipid, you provide an antigen, a place where an antibody can bind. So blood type A people have antibodies against the B carbohydrates. Blood type B people have antibodies against the A carbohydrates. And blood type O don't have antibodies against either A or B. So the blood type O, which is not modified with carbohydrate can give blood to any blood type because they're not supplying any antigens. They're not supplying, because there's no carbohydrates on the outside of that blood type, then there is nothing for an antibody in the receiver to attach to because there's no carbohydrates there. That would be O negative. So your plus type is just like another type of carbohydrate in addition to AB. And there are lots more. If you get into, say, you're considering donating an organ to a recipient, like a kidney or something like that, they would do a much more extensive typing of your tissues to see what kind of antigens you have present and if you're compatible with the person you're trying to donate to. Okay, when there is carbohydrate modification on a protein, that carbohydrate is almost always on the extracellular face, not the cytoplasmic face. And that's also true if the carbohydrate modification is on the phospholipid too. So on the face, facing the blood, if it is an organism that has blood, and not on the face facing the cytoplasm. There are different kinds of sugars that can modify phospholipids and proteins that are stuck in membrane. We'll talk a lot about this one, mannose, which is added on in the Golgi apparatus after a protein is synthesized and is important for signaling where that protein has to go and its eventual resting place in the cell and where it's going to function. So to review on phospholipids, you have phospholipid diversity because of the head group. You have phospholipid diversity because of the acyl chains. You have phospholipid diversity because they may or may not be glycosylated, have a carbohydrate added to them, and you have diversity in that the type of carbohydrate and the number of carbohydrates added to them. You also have sterols that are important in membrane function. Cholesterol is the one is shown here, and that's the one that we're mostly going to talk about. It gets embedded between individual phospholipids in a membrane, and it can adjust that membrane's fluidity. So membrane fluidity is a big feature of membrane function. Membranes must remain fluid in that all the components in that fluid mosaic membrane have to be able to move. So I often ask the question on a test, in the fluid mosaic membrane, what is fluid and what is mosaic? So fluid means that all the parts move, the proteins that are embedded in the membrane move, and the phospholipids move. And the mosaic means that it's made up of different kinds of molecules, lipids, proteins, carbohydrates, all make up the membrane. Okay, if we're just talking again about the phospholipids, again, here's the head group, here are the acyl chain tails. The tail degree of saturation, if they are saturated, remember, that means they have the maximum number of hydrogens possible. If they are fully saturated, the chain will be straight, and it's easy for these chains to stack up together. And if they are unsaturated, either one or more double bonds, that puts a kink in the tail. It's very hard to pack these together. And so this will lead to a more fluid membrane. This will lead to a less fluid membrane. This fluidity of the membrane has important health effects for humans. And one of the ideas has been that the more that your lipids look like this, the easier they are to stack together. And they can stack together also outside your cells, like in your arteries, and cause blockages. And with that idea, the idea of omega-3 fatty acids that are polyunsaturated means that they have more than one double bond in their chain. they became really popular. They're called omega-3 because at least one of those bonds is on the number 3 carbon, counting from the top of the chain, the carboxyl end of the chain toward the end. Omega-3 fatty acids, you may have heard about them in the news. You may even see supplements like this in the grocery store. They're considered heart healthy, and the reason for that was that native populations that live at high latitudes, so these are Eskimos that live in Greenland, they have a high-fat diet in that they're eating animals that live at cold body temperature. Cold body temperature animals have a lot of fat, and there are also not a lot of vegetables around to eat. So they have a very high-fat diet, which you normally think of as bad in terms of accumulating lipids in your arteries. But these populations have very low incidence of heart disease. So they thought it was because that the type of fat they're eating was high in omega-3 fatty acids. These are unsaturated, more this sort of shape of fatty acid, and so not as easy to clump up in your arteries, and they thought that's what made them heart healthy. But in 2015, there was a study done that this population, this human population, has an enzyme that is a desaturase that can take apart these omega-3 fatty acids, and that Europeans, like most of the people in this country, or maybe not most anymore, but most of the people where they have data from this country anyway, they don't have this desaturase. And so the assumption was that all humans are the same and that all humans could benefit from these omega-3 fatty acids, but there is no evidence in Europeans that omega-3 supplements reduce heart disease, or that even people that are of European descent eating a lot of omega-3 fatty acids in their diet can even digest those fatty acids. So interesting little bit of science there. Okay, so the straightness or the saturation of the fatty acid tails in a phospholipid depends on, has consequences for how stiff that membrane is going to be. And how stiff that membrane is going to be is related to when it's going to melt. Think of a stick of butter, and that stick of butter has a different composition in terms of it being in your fridge where it's really stiff. If it's on your countertop, it's still a solid, but just barely so. And then if you put it in a frying pan, it melts. The more unsaturated fats in that membrane or in that stick of butter, the lower temperature in which it will melt. So if you have more saturated fat in that membrane, it melts at a higher temperature. So as an example of stearic acid, which is 18 carbons with no double bonds, it has a melting temperature of 70 degrees C, which is really high. So it'll stay solid until it gets pretty warm. And this would be like in beef fat. You would see high concentration of stearic acid. Whereas if you go to oleic acid, which is the same number of carbons in the chain, but you just add one double bond to it, it melts at a much lower temperature. So one way you can adjust your membrane fluidity is just by adjusting the number of bonds in your phospholipids. It's not just bonds, although bonds has a big effect. So if we look at the melting temperature of something that is 18 carbons long, stearic acid, and no double bonds, it has a very high melting temperature. But if you had just one double bond, it goes down to about 16 degrees centigrade as a melting temperature. And if you add a second double bond, you don't get a big effect, as big an effect. So you get most of your effect from the first double bond, and adding a second one or a third one you do get an effect, but just not as much of an effect. So you get most of that kink, most of that lack of stacking from the first double bond. It's not the only thing that affects fluidity. Um, if you have a longer fatty acid chain, it'll be less fluid than a shorter fatty acid chain without the effects of the double bond, but you get the biggest change when you add that first double bond. Okay, so in this H5P question, you're going to have to first look at this slide and then answer the question, and hopefully you can answer that based on a clue I gave you before about another word for phospholipid. Okay, another thing that you've probably heard about in talking about fats in the news is cis fat versus trans fat. And you may know that trans fats are bad, but why are they bad? So in the cis conformation, when you add a double bond to a fatty acid, the chain going into the double bond and the chain coming off of the double bond come off on the same side of the chain. In the trans conformation, the chain going in and the chain going out come off on opposite sides of the chain. This has the effect of this conformation being straighter, not straight, but straighter, and this one being more kinked. So trans fats are more easily stacked together and can stay solid at a higher temperature. And they're also not seen a whole lot in nature. So when you do see trans fats is as a result of partially hydrogenating vegetable oil. So you take vegetable oil that has double bonds in it and you add hydrogens back to them, partially hydrogenating it chemically, and you get trans fats out of that process. And because trans fats, even though they have a double bond in them, are straighter than cis fats, they can stack together and cause heart disease because of plaques forming in your arteries. But also, they're much harder to get rid of if you are trying to metabolize those because your enzymes are not recognizing that shape. So the enzymes that would take apart a fatty acid don't recognize this shape as well as that shape. Trans fats are now banned in many states. Cholesterol. Cholesterol is another thing that you probably heard about in that at high temperatures, adding cholesterol increases membrane stiffness. At low temperatures, adding cholesterol decreases membrane stiffness. As I said when we talked about lipids in another podcast, you can't have a membrane without cholesterol. So you need it. But if you have too much of it, outside of what you need for cells, it is a sticky lipid molecule and it will contribute to those large plaques that can form in arteries. So you need cholesterol in your diet, you just don't need, you need the right amount. And how cholesterol affects membranes is a little bit complicated. So if you are a low temperature organism like a fish, low body temperature, and you add cholesterol to your membranes, it prevents your membranes from being too loose. If you're a high temperature organism like a mammal and you add cholesterol to your membrane, it increases your membrane stiffness. So mammals adding cholesterol increases stiffness in the membranes, prevents them from being too loose. And fish adding cholesterol to the membrane decreases stiffness of the membrane. Adding cholesterol makes them more loose. And by adding, subtracting cholesterol, an organism can adjust its membrane stiffness to be just right. If you use a technique called atomic force microscopy, where you drag a very sensitive needle across the surface of things, you can start getting a picture of what is called these lipid rafts. So the membrane is, the components of the membrane are constantly moving, and the phospholipids are not uniformly distributed throughout the membrane. They form these little rafts typically around a protein where it might be enriched with one kind of phospholipid, for example, phosphatidylcholine, and higher concentration in the raft than outside the raft. Even though if you extracted the whole membrane, you might get a different concentration. In these rafts, they can concentrate certain kinds of phospholipids over other kinds. And that can be important functionally because it could be that an enzyme that is dependent on a certain amount of flexibility or dependent on interaction with a certain phospholipid might have a better function within a raft than outside a raft. So it's a way for a membrane to be heterogeneous within the membrane. So even though everything is moving around, proteins can recruit specific phospholipids around them, and that can affect their function, and that's called a lipid raft. So some evidence as to how proteins move in the membrane and the fact that there are lots of proteins in the membrane, different techniques that show this. There's a technique called freeze fracture where you take a cell and you cut it and then you break it. First you freeze it and then you break it. And the frozen cell, when it's broken, tends to break down the middle of the bilayer, so the top half of the bilayer versus the bottom half of the bilayer. So these would be phospholipids sticking out. And then you see bumps. Then you would coat the top or the bottom half of this frozen piece with some kind of metal so that you could see it in an electron microscope. And then you see bumps where the proteins were when you froze it. Here's an example of this from a red blood cell membrane, and all these bumps are, you're looking at just half of the membrane, and you're looking at where the protein was. If you see a hole, and if you see like here, if you see a bump sticking out, that is where the protein is. So this is a protein that was in the other half and got stuck, and this is a protein in this half that got stuck. But the point is, there's lots and lots of proteins in this membrane. As we mentioned before, when we're talking about the fluid mosaic membranes, you can have these integral membrane proteins which pass through the membrane. They can pass through the membrane several times. You can have proteins that are associated just with one half of the membrane, say just the inner leaflet and not the outer leaflet. They don't poke out on the other side. You can have some that are just associated with the membrane that they don't actually are not part of the membrane, but they just stick next to the membrane. Sometimes they have some kind of anchor that is hydrophobic that associated with the membrane, but that's not the same as going through the membrane. There's all kinds of diversity in how proteins are associated with membranes. In the phospholipid part of the fluid mosaic model, the phospholipid themselves move, and they can move across one plane in a membrane. They can rotate on their axis, and they can flip from one surface to the next. This is the least common kind of movement. This is governed by a protein called flipase, but it does happen. These kinds of movement, rotating on its axis and lateral diffusion, are much more common and important to the function of the membrane. Some evidence that we know that this happens comes from a technique called FRAP, which stands for fluorescence recovery after photobleaching. So you label a whole bunch of membrane proteins with green fluorescence, and then you extinguish that fluorescence by hitting it with a really powerful light, and it bleaches out the fluorescence. So at that point, there are no fluorescent proteins, and if the proteins don't ever move, you should see that hole persist. But instead, as the membrane proteins are constantly moving, they will fill in that hole relatively quickly. So this is what the cell would look like, all those different proteins in the membrane before you bleach it. You zap it with a laser right at that point. There are no labeled proteins at that point, okay? There's still proteins there. Their label got bleached out by the laser, and then you can see that 20 seconds later, and then 100 seconds later, you can't even see where that was. So this proves that proteins are moving laterally in the membrane. Another experiment that has shown this is from a fusion experiment. So in cell culture, sometimes immortal cells, cancer cells, are fused with other cells in order to be able to grow them in culture. And you make what's called a hybridoma. So you can take a mouse cell with mouse proteins and a human cell with human proteins. You can label them different colors and then fuse their membranes. At first you see the mouse proteins on one side and the human proteins on the other side, and then eventually they're all mixed together. And again, this is evidence that proteins move laterally through the membrane. And then these experiments, again, this is the freeze-fracture sort of experiment. If you look at what the surface of this cell looks like, and all the bumps before you apply electrical current. After you apply electrical current, some of these proteins have a charge on them and they will move in response. So if they're negatively charged proteins and you apply a positive current on this side, they'll move to that side, and they can move because of the fluid mosaic membrane. They can move laterally within the membrane. If you have a fluid mosaic membrane that has all kinds of proteins in there, there are some proteins that you want to sort of stay put because they're going to be part of a signaling pathway that is going to signal something inside the cell. And so it's advantageous to have them be at a reliable place in the cell because of their interactions with something inside the cell. And you can think of this as like a buoy in the harbor. The water is flowing around the buoy, but the buoy is anchored to the ground. So for example, some of these proteins in a red blood cell called band 3 are anchored to other proteins that are part of the cytoskeletal network of the protein network that forms like a skeleton almost sort of inside the cell. And since they're attached to the inside of the cell, the fluid membrane can move around those, but they stay relatively in the same position. Certainly not true for all the proteins, because I just showed you proteins moving, but some proteins will stay put because they're attached to an anchor. Okay, here is a tricky question. I want you to go back and look at what we said about the effects of cholesterol on a warm-blooded animal versus a cold-blooded animal before you try to answer that question.